Good morning, ladies and gentlemen. Welcome to Biosimilars Workshop 2023. I am Pooja Sawant, the host for this session. We will begin with the first session, which is titled as Cell Line and Upstream Process Development. This session is sponsored by High Media. The moderator for this session is Dr. Sanjeev Gupta. Dr. Sanjeev Gupta has obtained his MSc in Applied Microbiology and Biotechnology and PhD in Molecular Biology and Cell Line Engineering. His core expertise lies in molecular biology, cell line development, upstream process development, bioanalytics, regulatory, as well as tech transfer to the GMP facilities for clinical and commercial manufacturing. He has published over 12 research articles, 6 book chapters, and has a patent to his name for biosimilar development. He has delivered talks on biosimilars in various national and international conferences. Dr. Gupta carries around 22 years of industrial experience and has been working since the year 2000 on the development of biosimilars, including monoclonal antibodies. Till date, he has developed and contributed over 14 recombinant molecules, out of which 8 biosimilars are already launched in the market, and the rest are in the developmental or clinical phase and are expected to be launched in the near future. I would request Dr. Sanjeev Gupta to take over. Thank you so much.
Thank you very much, Pooja, for kind introduction. A very good morning to everyone, and a warm welcome to the cell line development and upstream process event. This is the second series of today's session. We'll talk about the cell line development and process development. So we have distinguished speakers having a lot of experience in this area, cell line and process development. So they will be talking about some of the case studies, their experience, and the scale-up challenges, which we discuss during the panel discussion also. So as you know that cell line development, cell line and process, these are the key. That is the starting point for any biopharmaceutical development. It's regardless to say that these are the bedrock and decides fate of any biosimilar development, any biological development. So we face a lot of challenges when we talk about cell line, like the stability issues, then tighter expression. People talk about the expression, whether we can get 5 gram, 10 gram, and so forth. And with respect to process development, a lot of challenges when we talk about the consistency of the result. That is very important, because we struggle a lot on the consistency and to get the consistent result with respect to titer and quality of the products. So we will deep dive into these areas today. We have 3 talks today. One of the speakers, Dr. C. Prakash, is not coming today because of certain personal issues. He cannot come here. So we will be having 3 talks, 3 speakers, and after each talk, we'll have Q&A session. So we'll be given 3 to 5 minutes to ask the question. And followed by, we'll be having the panel discussion. After panel discussion, we'll give chance the audience to ask the question based on depends on the time we have. So let's start the session with the first speaker. I am pleased to invite our first speaker, Dr. Ravi Mishra. He is working as a principal investigator in CSIR-IMTECH, Chandigarh. Dr. Ravi has obtained his PhD from Banaras Hindu University. His expertise lies into the vaccine and process development of biotherapeutics. He carries overall 15 years of experience, both industry and academia. He has been a part of the discovery and development of protein-based vaccine against Staphylococcus aureus. He also developed next generation protein subunit-based vaccine against COVID-19. In the first session, we heard a lot about mRNA vaccine. So he also has expertise in the vaccine development. He's currently the principal. He's currently heading and principal as a principal investigator in CSIR Institute. And his title of the presentation is Emerging Cell Line Engineering Technologies and Their Potential for High Yielding Biopharmaceutical Production. So over to Dr. Ravi. Welcome you.
Okay.
Thanks.
Thank you. Thank you. Thank you, Sanjeev, for your kind words. So, just now I came to know that the previous speaker is not coming, who was supposed to discuss basics about cell line engineering, mostly CRISPR and other technology. So, I was told that to keep that aspect very brief. So, I will be not touching much of the basic cell line engineering aspect, but rather I will go to a case study which we have done with some basic engineering approach and demonstrated the production of one of viral protein spike. So I will be speaking more about that part. All right, so this is how I am going to navigate the presentation, starting from a brief introduction of biopharma, cell factories, outline of cell line process and engineering approach, and then I will come more detail into the case study for developing a CHO cell-based spike protein. So we have used a very unique approach to express spike protein, full-length spike protein. As a biopharma person, you might be knowing that expressing full-length spike protein is not a really easy task. So there was a lot of engineering at the level of molecule, at the level of cell line. All those things have done, and finally we now reach to demonstrated our data at hamster, and we are planning to take into the clinical maybe in few months as a kind of a new subunit protein vaccine. There is another very important initiative which I am leading at CSIR-IMTECH. It's a very unique program funded by National Biopharma Mission VIRAC. And this program is to set up a new facility. It's a first of its kind facility in India, it's going to be, which will be dedicated for addressing the cell banking need of the country and also outside. And this facility is going to provide biopharmaceutically relevant cell banks for the early development to the industry, particularly emerging industry startups. So it's a very big program where we have about 4,000 square feet area, clean room. We already have commissioned that facility. Now other things, activities going on. So this facility will be functional maybe in 3 to 4 months. Okay, so start with the biopharma, need not much detail, but biopharmaceutical broadly covers prophylactic and therapeutic, as you all know, which is diverse class vaccine, recombinant therapeutic protein like MABs, cell and gene therapy products, blood and blood component, hormones, anti-infectives. This I would like to this is a very interesting slide, which when I was working in GSK in Siena, Italy, my boss, his name is Rino Ropli. So when there was induction, he used to present this slide. So when I left GSK, I asked him, can you give me this slide? So this is a very interesting slide, particularly to explain how biopharmaceutical development happens. So what we call it the product development process. So it's a complex, integrated, structured, at the same time, flexible approach for the product development. So it starts with, the innovation phase, where you have 100,000 of idea for the molecule, different hypothesis based on the basic applied research, based on the, the need, medical need. It's different idea, hypothesis, and from industry perspective, the business opportunity. So you start with the multiple, I think this laser will not work here. Okay, that's fine. Okay, so you start with multiple idea, multiple molecule at the same time, and once it moves towards the, the funnel, you define the product. So you end up with the few molecule at the stage of definition. And slowly, when you move the development phase, in the preclinical phase, clinical phase, at the end you end up with the launching 1 or 2 molecule. And this is a very much regulated, once you enter in the definition phase, it's a very much regulated activity, particularly in industry, what we call toll gate model, where you have at each and every gate you have go-no-go decision taking matrix, and that is how we structure the program. To look at what is the broad process of the development of biopharma, starting from preclinical, which is early development process, and then you go to the clinical phase, and then finally launch. So this is a very broad overview of where you start and where you end. So coming to the recombinant protein expression machineries, what we call the cell factories. So I don't think we need much discussion on that, but the routinely used systems are here, and microbial variety of E. coli strains, Pseudomonas fluorescens, lactase, subtilis, glutamicum. So these are some strains which are either have some product associated with it or having something in the clinical. Each strain everyone know, Pichia pastoris, cerevisiae, Hansenella parvarfa, and so on and so forth. In eukaryotic side, you have, these are the common cell lines which are used for the production, sp2, ns0, when you come to hamster, CHO, BHK, human cell lines, HEC, and PERC6. And this particular use of the cell line and the choice of cell line depends on different kind of expectation from industry. Like what's the throughput of it, what kind of productivity this cell line may have, how this cell line can perform at manufacturing scale, how can you scale it up to take it larger scale, cost effectivity of course, safety and efficacy. These are the different approach. I will just go very briefly because I was assuming when I discussed with Sanjeev that the previous speaker was going to speak some of these detail. So I thought of just keeping a very brief outline. So these are the different approach which are used when we do any cell line engineering. So approaches like we engineer the cell proliferation and survival engineering, apoptosis regulation, cell cycle regulation, metabolic engineering, transcription optimization, translation, codon optimization. So at every stage there is a lot of, and these topics, one topic you can spend hours to discuss. So I cannot go much detail in that, but just I listed these kind of approach used to develop high producing viable cell lines. To execute these, we use different tools. Of course, there is conventional tool already there, which is commonly used for the random integration of the gene of interest in the genome, which is commonly. Now, the emerging tools are based on the genome editing, putting targeted integration in the genome using the recombination mediated cassettes, endonucleases. We use some allelic replacement subtle vectors which can propagate in the microbe at the same time in mammalian system, like in one vector which we have in our lab, which it multiplies at 30 degree in E. coli. When you put it 42 degree, it integrates in the genome. So we are adapting it to work in the mammalian system. So we cannot go 42 degree in mammalian system. So we are trying to tweak that vector. So that approach is also where you take you first identify the region where you want to integrate your gene of interest. So as we are discussing with Sanjeev, a lot of things are now happening to identify a place where you can integrate your gene. And that one of the important thing is that portion of the genome should be highly transcriptionally active, where you can put your gene, and that may give a higher thing. So first to identify the site where you want to integrate your thing, And that should not be essential to the cell line, to the particular cell. So you have to identify based on the genomics and proteomic data, identify the portion that where you need to integrate, take the up and down stream, almost 1 kb region of that particular location where you want to integrate, put in your vector, and by homologous recombination, when you switch the temperature for 30 to the higher temperature and integration happen, and there is a classical Mendelian, genetic mechanism happen. There will be first crossover, then when you will passage the cell, it will be second crossover. Ultimately, your gene will replace to that, and it will be fixed in the genome. CRISPR, again, it's an emerging tool, microRNA, siRNA, to engineer the glycosylation, glycotransferases. This kind of things are being used. These are the broad strategy used for the process development when you go to, when your aim is to improve yield, at the same time keeping the impurity profile in the limit and all those other parameters in the control. So you can do cell line, at the cell line level, metabolism engineering, protein secretion engineering, glycosylation. At the media and feed level, different media can be tried, different feed combinations, percentage of feed, media additive, etc. For the process optimization, you go batch, fed batch, perfusion. That is the emerging thing now. At the bioreactor level, you can play with the impeller type, stirring speed, sparger type, gas flow, and other parameters. So now I will come to one of our learning and experience, what we have with the developing a prefusion stabilized recombinant full-length spike protein in CHO cell. So we started this program in late 2020, and that time, a lot of number of vaccines were in pipeline. So we have been asked to initiate a program in our institute, and in order to set up a platform. So we initiated, and in our project monitoring board, I presented this, that I want to go with this approach. So they said that looks too complicated title. So you need to explain it. Why? Each and everything you need to explain it, then only we can think of giving you funding. So okay, then I have been asked different explanations why. Why spike? Why full length? Why not RVD? Everyone was doing. Why spike? Why prefusion state? Why you need to stabilize this protein in prefusion state? Why CHO cell, not microbial cells? And why protein subunit? There are a lot of number of different kind of approach now, like viral vectors, mRNA, all that. So these were the question asked with the our management, and scientifically how you explain this. So to be a good vaccine candidate, everyone knows that's a single rule. The molecule should be on the surface. I think everyone agree about that. So spike, why spike? Because it is on surface. And this very recent paper came in Nature. You can appreciate, it's a beautiful paper, you must read it. They have mapped how this spike protein is interacting with host receptor. What kind of nanosecond movement is happening in spike. You can see there's a trimeric spike protein because it is present in the trimeric state, and there is movement in the RBD. It comes in the up and down state. So when it comes in the up state, it interacts with AS2. So this is how it is searching the AS2 receptor. This is what this virus does. So having fully decorated spike on the surface, and this is how it interacts. So why is spike? Answer is clear, it's on surface. It is the major virulence factor. Why full length? Why not RBD? A lot of debate was there because full length is too bulky. I can tell you spike is not a single protein, it's the complete protein machinery. It's a type 1 toxin. Everyone who works on this kind of protein, you know that it's a complete machinery, a very dynamic protein. So we asked why you want to go with the full length? It's too difficult. It's very difficult to express. And this was one of the analysis I did at that time. As you know that most of the mutations are located in the RBD region. You can look at all those variant of concern, alpha, beta, gamma, delta, delta plus, omicron is also there. Most of the mutations were on the so it's a risky, with what we thought scientifically, it may be risky to have this just RBD, and we thought that maybe there may be some other receptor which might be playing role in the interaction with the host. So better to take the full length. And also there was another point which we thought of, because if RBD is the only master major portion which is interacting with the host, why a virus will make a protein of 600 kD, 180 monomer? Yeah. So why prefusion? Because of the prefusion stabilized is as I said, that's the meta-stable protein. It goes conformational changes from prefusion state to postfusion state. And to use that as an antigen, you need to lock this protein in prefusion state. And that's where you do all the protein structural biology and protein engineering approach to lock the protein in prefusion state, because in the prefusion state is the antigenic state and postfusion is the purely antigenic. So once it goes from the prefusion to postfusion, there is a massive rearrangement you can see in the different epitopes, and it's not a good antigen. So you need to express protein, sterilize protein prefusion state. Why CHO? Answer is simple, because it's one of the most commonly used, all the impurity profiles, density, high cell density you can cultivate, and so many regions. So, and why protein? So our logic was that we wanted to work on the protein, because we know that protein-based vaccine are known for having excellent safety and efficacy record. Protein-based vaccine is considered one of the most effective. This topic may be debatable, but there are many. So robust immunogenicity and memory response. My colleague Nimesh Gupta is there, he may put some light on that. But yeah, so these were the rationale which we have proposed, that why we want to use protein-based approach. And as you know, that protein-based vaccine can be used from all ages. Like for example, hepatitis B from maybe a week after birth to different ages. Okay, so we designed the antigen using structural biology and protein stabilization approach, using different criteria here mentioned here. So this protein is having two major domains, S1 and S2, and S1 and S2 is cleaved during the interaction. There is a site called a furin cleavage site. So you need to modify that site. We put some mutations to increase the loop stability. We inserted some proline residues. We introduced the disulfide bridges. I'm not going very much detail of it because it's a part of one of the IP which we have submitted. But there are specific residues which we have touched, and we generated a dozen of different antigens. We changed amino acid to introduce the salt bridge. Cavity filling, we increased the chain length of certain amino acid to do the cavity filling. We screen several secretory signals, because there is a lot of impact of what kind of secretory signal you are using, I can tell you. Because in one of our case we get almost 1 gram of the yield, but when we look back in mRNA level in the cell, you have get a lot of RNA is present. So you need to tune the secretory signal. Removal of transmembrane domain, which was one of the factors destabilizing the this protein. So this is how we did our development. First, we cloned this engineered sequence in the pcDNA and demonstrated the transient expression. Once we got the expression confirmed, we have transferred this gene to a production vector, which is industry compatible, the Freedom PCHO. And from there, we confirm with the basic molecular biology that the integration is there. And this CHO-S cell line, which we have, is a DHFR negative. We use PCHO vector, which is positive. we use a hybrid promoter. We did unique codon optimization, sequence engineering, security signal as I mentioned, and finally we developed stable cell line. Here you can see this was one of the very exciting moment for us when we did, and this is a 10 microliter of the supernatant directly. And you can see the expression of full length, and not only the expression, but protein looks like quite stable. From there, we moved to the developing stable cell line, where we used the standard procedure. Since we are using a thermo kit, so we followed this with some modification in the process, which broadly in the transfection stage, then selection phase 1, selection phase 2, finally we end up with 4 different cell pool, which is stable cell pool. And this is the pre-transfection profile, so you need to give 5 to 6 passage before you transfect it. And once you transfect and put the selection pressure, viability will go down. It's a very standard thing. And slowly it will recover, then you put the further selection pressure, and finally you will get the stable cell line. Right, so we got 15 stable cell pool generated, and then we evaluated just a safe class batch and fed batch mode expression. In the left you have the VCD versus viability, and then glucose consumption versus the metabolite. In the down you have, so you can see that it's at the day 7 we are getting almost 200 mg per liter. And at that point of time, I remember I procured some for the reference, and that molecule was something like 100 microgram, was something like Indian currency 1.5 lakh. So we got a lot of, so this, and also in literature, a lot of people have reported that it's very difficult to express. So you go in mg in a few, mgs. So it was a very good thing for us. And then we, optimized the fed batch. We got further enhancement in the yield, close to 300. Then we did the bioreactor optimization at small scale bioreactor. And this is what parameters we have used. We have used BioFlow 320, 1 liter volume, and different parameters mentioned here we used to scale our antigen in the bioreactor, and overall, the cell culture was for 12 days. This is profile of bioreactor run, and finally we reached to the almost 500 mg per liter. And this is for the 1 liter bioreactor. And so we had enough protein to play around for the analytical and preclinical. So this is the profile as day space, and western blot you can see the antigen is quite stable, and you can clearly see a single band. And antigen is the important in the size extended HPLC. You can see it's almost 600 kD, which correspond to the trimeric state of spike, which is present basically on the viral surface, because we engineer the sequence in such a way that it will make a trimer. Okay, so this was good, but we wanted to see whether it is really looking like trimer, and this was the data. So when we did the negative stain microscopy, we can see that in the left one is the spike. It's a cartoon for a spike trimer, and here is our molecule. So this, we stopped there. We have the molecule which is looking like the trimer, and it's quite stable. Then we evaluated the vaccine efficacy in preclinical model. In the left is the mouse ELISA, excellent immunogenicity. In vitro neutralization across different variant of concern which were present at that point of time. You can see there is almost very small difference from different variants. This was very, very important point. And the hamster data. Almost we are getting sterilizing immunity here. In the top is our data where we get almost 5 log reduction in the viral load. And this is one of the data published from Covishield. So we are non-inferior to that data. So this all overall story was covered in couple of patents we have filed. In collaboration with Dr. Nimesh Gupta from NII. And we get good germinal center response. So if there is, so I'm not immunologist. If you have any question, that is the person you can ask him. But looks like vaccine is also inducing that particular arm of the immunity. All right, I will not go to that. Okay, this is so, as I mentioned before, that biopharmaceutical development is a very structured and controlled way. So this is how we move to our development. Now this proof of concept data is ready for the regulatory development. Very brief, as I said, that we are setting up a facility of national importance at CSIR-IMTECH with the support of National Biopharma Mission. And this facility is going to be India's first facility which will help industry for development of the cell bank of biopharma relevance. This facility, mostly we cover microbial cell hosts which are used for development of biopharma, expression vectors, characterization of the cell bank as per ICS guideline, and a small scale technology proof of concept can be generated using our facility to different industry who can take this their molecule further. And this is all those forwarded. So we have almost 4,000 square feet area where the facility is set up. So maybe in a couple of months we will inaugurate that. Okay, thank you. So this is most important. This is my team. So all the work I am presenting on their behalf. And the funding support from CSIR, my institute, BIRAC, the fellowship to my students, my collaborators, Rajesh Ringe, Nimesh Gupta, Sasank Tripathi from IISc for Hamster, Dr. Ram Vishwakarma, our Director General, and Director Sanjeev Khoshla for all the support. Thank you very much.
Thank you very much, Dr. Ravi, for such insightful talk. I really appreciate the research work which we are doing. You are doing novel research, and this is more towards applied. It's very rare in India when you talk about the academia and industry. So it's really very good piece of work which you are doing, right from the clone to process scale up and proof of concept up to preclinical trial. And it's very interesting that you are setting up a new facility for the cell banking and cell bank characterization in India, which is not available in India. So thank you very much. Let's give a round of applause for Dr. Ravi. Thanks. Okay. So now session is open for the question and answer. We can take 2 questions. Yeah. So, yeah. So please introduce yourself, your name and organization name.
Hi, Dr. Mishra. It was a very interesting topic, not at all boring. And I'm Abhishek Naik from VJ Bio. I have a question, would like to know what are the critical process parameters to look into to have a stable cell line after a post-transfection.
So the thing is that it's a very delicate, as you know, that's very delicate, and you cannot be get success in one attempt. My student who did all this work is there, so I was shouting him all the time because there was a huge pressure. So the first thing you have to handle the things in a very delicate way. That although they mentioned that you need to use 100 microgram of pyromycin and all that thing, but you need to set up your titration first. It's not that it will work in your system. So you may go lower. First you adapt the cell. And so that is one. Go slowly, do the titration with a small amount. And then you move further to the things. And then you need to monitor the viability very closely. So that is one parameter which you should also consider that. But the thing is that the selection pressure is the point where you need to tweak or you need to titrate it.
Good afternoon, doctor. Myself Sumit More. I am representing Serum Institute of India. So my one question is that I want to add one more why. Why you are not using the HEK cell line instead of CHO?
The simple question is, we for the CHO we had the licensed CHO, so we used that. We tried also in HEK, And when we compared the expression profile in HEK, this particular thing was much inferior than CHO.
Okay, and what about the
But-
purity?
Purity was
and the potency?
okay. Purity profile, there was no problem, but expression was much inferior in HEK
Okay.
than CHO S.
Yeah, because the glycosylation pattern of HEK versus the CHO, both are the different.
Yeah, but we looked the glycosylation profile broadly, and we are getting glycosylation which is desired. So we have the human-like glycosylation in this system as well,
Okay.
and it is not a normal antibody where you have one or two. It's a 22 sites where glycosylation is there.
Okay.
So a massive glycosylation, and also in this case, what literature said that glycosylation is there, but it doesn't play much role in the immunogenicity or in the the further protection. So that you can if there is also inferior glycosylation or not matching the what theoretically is, antigen is quite well.
Okay, and have you compared this subunit vaccine against the commercialized Covax? Because
We are
it's
doing
also-
now. We are doing now.
Okay.
Yeah.
Thank you.
Thank you.
You can take one more question, Dr. Patil.
Yeah. Hi, Dr. Ravi. It was really a nice presentation. My question is related to designing of spike protein. So basically, how big stretch of amino acid you have added in that, and whether you have done in silico validation that whether it, because you are making it for making some novel vaccines which is more potent. So how do you know that it is not immunogenic in nature? Because you have added some stretch of some
Yeah.
10-15 amino acid in that.
So it's not very, it's not 10-15 amino acid. The things I mentioned are the principle what we did. So the construct which we have where we have done 6 to 7 point mutation, single amino acid, not the big stretch.
Okay.
So, but we have multiple construct. First we screened so many construct using different approach. So one of the approach which worked was putting few prolines and modifying the furin cleavage site, removing significant part of the C-terminal region which was destabilizing when you do the normal mode analysis. So we do first in silico, molecular dynamics simulation and all that, and based on that data, we decide how to go.
So have you compared the immunogenicity of wild protein, spike protein, and the engineered protein, that it has the similar immunogenicity or very different or minimum or maximum?
Spike-
that data.
We did not get any reference material because we got some, I cannot name, some full length on the name of full length spike, but within 2 days it was smear. So the thing is, if you want to express a spike as a recombinant full length, you need to engineer it. So you don't have any way to express it. It will not be expressed as wild type in the full length.
Yeah, you get either S1 or S2 in the market, or
Yeah.
either RBD. Yeah.
It's very tricky, and then so there was, but you need to look at the end point. So when you go in hamster, we get all the histopathological features and everything is safety and immunogenicity and protection.
Okay.
So that's what is the most important thing.
Thank you.
Thank you.
I think we are running short of time. Maybe we can take a question later. You can interact with Dr. Ravi later on, maybe during lunch also. So thank you very much, Dr. Ravi.